And the question is, microbes are present in soil, thermal vents, polluted water, or all of these? And the answer for this is all of these. That is soil, thermal vents, and polluted water. Let's know how. Microbes are omnipresent, found in soil, water, oil, ice, inside bodies of human beings, animals, and plants. Some are found in hot springs under 80 degrees Celsius to 100 degrees Celsius, and even in geysers, which is also called as thermal vents. Let's move on to the next question. Which of the following microbes is a proteinaceous infectious agent? Fungi, prions, bacteria, or protozoa? And the answer for this is prions. Let's know how. Let's know what is prions first. Prions are highly resistant glycoprotein. Highly resistant glycoprotein. Particles which function as infectious agents. Prions can also act as catalysts. They can also act as catalyst, converting normal protein into prion state. Prions are not affected by proteases. Let's note it down. Nucleases, temperature, which is particularly up to 800 degrees Celsius, UV radiations, or formaldehyde. So the answer for this is prions. Prions are highly resistant glycoprotein. They act as catalyst. They are highly resistant and not affected by proteases, nucleases, temperature, which is particularly up to 800 degrees Celsius, temperature, and UV radiations plus formaldehyde. Okay, this is an interesting question. Which of the following food items is produced by the fermenting activity of microbes? The options are idli, dosa, todi, or cheese. And the options next are here. Idli and todi. Todi is a drink. C, which is todi and cheese. C, idli, dosa, and todi, or all of these, that is idli, dosa, todi, or cheese. And the answer for this is all of these. Let's know what is cheese and how is it made. Cheese is one of the oldest milk products prepared with the help of microbes. The curd is separated from liquid part or when to form cheese. Dosa, upma, and idli are fermented preparation of rice and black gram. The two are allowed to ferment for 3 to 12 hours with air borne leuconostoc and streptococcus species of bacteria. These two are very important. If these two bacteria is not present in any type of fermentation food, then the fermenting activity of microbes cannot happen. So let's know what is todi. Todi is a traditional drink, as I said before, of some parts of South India like Tamil Nadu, Kerala, Karnataka, Andhra Pradesh, which is made by fermentation of sap of palms. And the answer for this is all of these, as we saw just right now: idli, dosa, todi, and cheese. Now, cheese is one of the oldest milk product prepared with the help of microbes. The curd is separated from liquid part or whey to form cheese. Dosa and upma and idli are fermented preparation of rice and black gram. Okay, the two are allowed to ferment for 3 to 12 hours with airborne. These two species of bacteria, that is leuconostic. You can separate it while pronouncing leuconostic and streptococcus. Yeah. And what is todi? Todi is a traditional drink which is done in some parts of South India, which is made by fermentation of sap of palms. Let's move on to the next question. Streptomycin is obtained from Streptomyces grassis, Streptomyces cerevisiae, Streptomyces venezuelae, or Streptomyces ramosis. And the answer for this is Streptomyces grassis. Let's know how. And let's know what is Streptomyces grassis. Streptomyces grassis is an enzyme obtained from the cultures of some hemolytic bacterium, that is Streptococcus, and modified genetically to function as clot buster. It has fibrinolytic effect. Therefore, it helps in clearing blood clots inside the blood vessels in patients who have undergone myocardial infarction. Myocardial infarction, which leads to heart attacks through dissolution of intravascular fibrin. So, streptomycin is obtained from Streptomyces grassis. This Streptomyces grassis functions as clot buster, and it is a dissolution of intravascular fibrin which leads to heart attacks through intravascular fibrin. And let's move on to the next question. Which of the following antibiotics was extensively used to treat American soldiers wounded in World War II? This is an important question. And the options are as follows: neomycin, bactitracin, chloramphenicol, or penicillin. And the answer for this is penicillin. Let's know how. In 1940, in the year 1940, E. Chain, these two scientists named E. Chain and H. Florey, obtained a relatively stable preparation of penicillin, which was extensively used to treat the American soldiers which were wounded in World War II. So always remember, penicillin was the antibiotic penicillin was the antibiotic used extensively in 1940, used to treat American soldiers who were wounded in World War II. Let's move on to the next question. Which of the following options contains the end product? The end product formed during anaerobic respiration in yeast. This is also an important question. And the options are H2O, CO2 and energy, H2S, C6, H12O6 and energy, CO2, C2, H5OH and energy, or H2O and CO2. And the right answer for this is CO2, C2, H5OH and energy. Let's know how. Anaerobic respiration in yeast results in the production of ethyl alcohol, carbon dioxide, which is CO2, and energy. So what is ethyl alcohol? Ethyl alcohol is itself C2H5OH, CO2 and energy. Let's write down the formula, and the formula goes this way. C6H12O6 with zymase and complex C6H12O6 gives with zymase and complex 2C2H2OH plus 2CO2 plus energy. And the formula is C6H12O6 with zymase and complex gives 2C2H5OH plus 2CO2 plus energy. Now this is the formula for anaerobic respiration in yeast. So the question was what is the end product formed during anaerobic respiration? This is the formula for anaerobic respiration of yeast, and the end products for these are C2H5OH, CO2 and energy. C2H5OH is ethyl alcohol, CO2 is carbon dioxide and also with energy. So the end product formed during anaerobic respiration yeast is option C. Let's move on to the next question. Probiotics are cancer-inducing microbes, new kind of food allergens, live microbial food supplement, or safe antibiotics. And the right answer for this is live microbial food supplement. First, let's know what are microbiotics. Microbiotics are live microorganisms, bacteria in most cases, that are similar to beneficial microorganisms found in the human gut. They are also microbiotics are also called as friendly bacteria. Let's know why. Friendly bacteria or also known as good bacteria. You may also get a question as probiotics are also called as friendly bacteria, good bacteria, bad bacteria, or option A and B. So you may tick mark option A and B, which is friendly and good bacteria. Let's come back to the answer. Probiotic microorganisms consist mostly of strains of Lactobacillus, Bifidobacterium, and Streptococcus. So probiotics are taken as food supplement and energy drinks. Yes, you may know this energy drink in which, yes, Shilpa Shetty acts in that ad, which is Yakult. So probiotics are live microbial food supplement which is present in energy drink, for example Yakult. These are also known as friendly bacteria and good bacteria, which is very good for your health, intestines, and also pancreas. Lactobacillus, Bifidobacterium, and Streptococcus are present in probiotics. And let's move on to the next question. Monoscus purpuris is a yeast commercially used in the production of, the options are citric acid, ethanol, blood cholesterol lowering statins, streptokinase for removing clots from blood vessels. And the answer for this is blood cholesterol lowering statins. Let's know how. Statins are products of fermentation by the yeast Monoscus purpuris, which resembles malvolate and are competitive inhibitors of beta hydroxy beta methyl glutanyl HMG CO antigen reductase. This inhibits cholesterol synthesis. Statins are therefore used in lowering blood cholesterol. And the examples are very, very important. Let's name it out. The first one is lovastatin. Next one is pravastatin. And the last one is simvastatin. Okay, Monoscus purpuris is a yeast commercially used in the production of blood cholesterol lowering statins, and beta hydroxy beta methyl glutanyl is present, and the very, very important examples are as follow, that is lovastatin, and simvastatin. And the next question is, which of the following steps is taken by the Ministry of Environment and Forests to protect rivers from water pollution? And the options are Ganga Action Plan, Narmada Action Plan, Yamuna Action Plan, or both A and B, that is Ganga and Narmada Action Plan. And the answer for this is both Ganga and Narmada Action Plan. The Ministry of Environment and Forests are initiated Ganga Action Plan and Yamuna Action Plan to save these major rivers of our country from pollution. Under these plans, it is proposed to build a large number of sewage treatment It is proposed to build a large number of sewage treatment plants so that only treated water may be discharged in the rivers. So Ganga and Narmada Action Plan were taken by the Ministry of Environment and Forests to protect rivers from water pollution. Let's move on to the next question. Which of the following antibiotics is used to cure throat infections? And the options are neomycin, polymyxis, tetracyclines or viridin. And the answer for this is polymyxis. Let's know how. Among the given antibiotics, that is neomycin, tetracyclines and viridin, tetracyclines auramycin obtained from Streptomyces aureus is used to cure whooping cough. Some of the antibiotics are also useful. For example, chloramphenicol and erythromycin. Let's move on to the next question. Enzyme which has the fibrinolytic effect is protease, amylase, lipase or streptokinase. And the answer for this is streptokinase. Streptokinase tissue plasminogen activator or TPA is an enzyme obtained from the cultures of some hemolytic bacterium. Streptococcus are modified genetically to function as clot buster. It has fibrinolytic effect, therefore it helps cleaning blood clots inside the blood vessels in patients who have undergone myocardial infarction, which leads to heart attacks through dissolution of intravascular fibrin. And let's move on to the next question. Dash is the first step of sewage treatment. The options are precipitation, chlorination, sedimentation or aeration. And the answer for this is sedimentation. Let's know how. Primary treatment in sewage treatment involves physical removal of particles, large and small, from the sewage. Removal of particles, large and small, from the sewage through filtration and sedimentation. through filtration and sedimentation. Through and filtration and sedimentation. Now, initially, floating debris is removed by sequential filtration. Then, the grit, soil, and small pebbles are removed by sedimentation. Always remember the first step of sewage treatment is or filtration. And the activated sludge system, a part of secondary treatment, is one of the widely used aerobic treatment systems for wastewater, in which very vigorous aeration of the sewage is done. The sewage is passed into an aeration tank from primary settling tank. Tank from primary settling tank. The flocks are allowed to settle down in secondary settling tank. In settling tank, the bacterial flocks are allowed to undergo sedimentation. The effluent of supernatant is generally passed into natural water bodies like rivers, ponds, streams, etc. The sediment of settling tank is called activated sludge. A part of it is used as inoculum is aeration tanks. The remaining part is passed into a large tank called anaerobic sludge digesters. Let's move on to the next question. The purpose of biological treatment of wastewater is to reduce BOD, increase BOD, reduce sedimentation, increase sedimentation. And the answer for this is it reduces the BOD. Let's know how. Secondary treatment is sewage of biological treatment. It depletes 90 to 95% of the BOD and many pathogens are removed. Reduction of BOD of 90% is achieved through mineralization of small fraction of organic matter and conversion of large proportion to removable solvents. And the next question is BOD is dash in polluted water and dash in potable water. Options are more, less, more, less in both, or medium in both. And the right answer is BOD is more in polluted water and less in potable water. And let's know how. Primary treatment in sewage treatment involves removal of particles, large and small, from the sewage through filtration. And the activated sludge system, a part of secondary treatment, is one of the widely used aerobic treatment systems for wastewater, in which very vigorous aeration of the sewage is done. The sewage is passed into an aeration tank from primary settling tank. The flocks are allowed to settle down in secondary settling tank. In secondary settling tank, the bacterial flocks are allowed to generally pass into natural water bodies like rivers or streams or ponds. So the sediment is a settling tank which is used as activated sludge. The part of it is used as inoculum in aeration tank. The remaining part is passed into a large tank called anaerobic sludge digester. So BOD is more in polluted water and less in potable water. Let's move on to the next question. Process of biogas production is anaerobic process, aerobic process, active process, or passive process. And the answer for this is B, which is anaerobic process. Let's know how. Now, what is biogas? Biogas is a methane-rich fuel gas produced by anaerobic breakdown of digestion of biomass with the help of methanogenic bacteria. Biogas is made up of methane, 50 to 70% carbon dioxide, 30% to 40%, with traces of NO2, H2SO, or H2O. Let's move on to the next question. These bacteria grow anaerobically on cellulosic material, produces large amount of methane along with CO2 and H2 and are collectively called as methanogens. Examples of such bacteria are Methanobacterium, Methanobrulacter, or Methanococcus, or all of these. And the right answer for this is three of them, that is Methanobacterium, Methanobrubacter, and Methanococcus. Let's know how. Methanogens are microorganisms that produces methane as a metabolic byproduct in ionoxic condition. They include Methanobacterium, Methanobrubacter, and Methanococcus. Let's move on to the next question. Bacillus thuringiensis or BT strains have been used for designing novel, and the options are as follows. You may read the question. Yes, and the answer for this is bioinsecticide plant. Let's know what are bioinsecticides. Bioinsecticides are those biological agents that are used to control harmful insects. Because of development of methods of genetic engineering, B. thuringiensis, toxic gases are introduced into plants. Such plants are resistant to attack by insect pests. PT cotton is one such example. Let's move on to the next question. Bacillus thuringiensis is used to control bacterial pathogens, fungal pathogens, nematodes, or insect pests. And the answer for this is D. And the right answer for this is D, that is insect pests. Now let's know how. Bacillus thuringiensis is used to control insect pests. Bioinsects are those biological agents that are used to control harmful insects. Because of development of methods of genetic engineering, Bacillus thuringiensis is toxic genes which are introduced into plants. Such plants are resistant to attack by insect pests, but cotton is one such example. Let's move on to the next question. Biopesticides are the chemicals which are used to destroy the pests, the living organisms or their products which are used for the pest control, the organisms which destroy the crops, or none of these. And the answer for this is B, which is the living organisms or their products which are used for the pest control. Let's know how. Biopesticides are those biological agents or their products that are used for control of weeds and insects, which are also called as pathogens. Most important example is the soil bacterium, which is pterogenesis. And the next question is microbial biocontrol agent that can be used to control butterfly caterpillars is Trichoderma polysporum, Bacillus thuringiensis, Streptococcus or mycorrhiza. And the answer for this is B, which is Bacillus thuringiensis. Let's know how. An example of microbial biocontrol agents that can be introduced in order to control butterfly caterpillars is the bacteria Bacillus thuringiensis, which is BT. These are available in sachets as dried spores which are mixed with water and sprayed onto vulnerable plants such as brassicas and fruit trees, where these are eaten by the insect larvae. In the tree's gut of the larva, the toxin is released and the larva gets killed. So, the bacterial disease will kill the bacterials and the caterpillars, but which leaves other insects unharmed. And the question is, which of the following is not used as a pesticide? And the options are as follows: Trichoderma harzianum, Nucleoporehydrovirus, Xanthomonas capistris, or Bacillus thuringiensis. And the answer for this is C, which is Xanthomonas campestris. Let's know how. Xanthomonas campestris is not used as a pesticide. Bacillus thuringiensis is used as a microbial biocontrol agent. Trichoderma harzianus is a free-living fungi, common in soil and root ecosystems. It is an effective biocontrol agent of several plant pathogens. Viruses of the family agent of several plant pathogens. Baculovirus are pathogens that attack insects and other arthropods. Arthropods such as cockroaches, and the majority of baculoviruses used as biological control agent belongs to the genus. This is extremely important. Nucleopolyhedrovirus. Nucleopolyhedrovirus. So, which is not used as a biopesticide? The answer is Xanthomonas campestris, not Trichoderma harzianum, Nucleopolyhedrovirus, Bacillus thuringiensis. Let's move on to the next question. Baculoviruses, nucleopolyhedrovirus do not show host specificity, narrow spectrum applications, effects on non-target applications, or utility in 1 p.m. program. And the answer for this is effects on non-target applications. Let's know how. Let's know how. Nucleopolyhedrovirus are excellent candidates for species-specific, narrow-spectrum insecticidal applications. They have been shown to have no negative impacts on plants, animals, birds, fish, or even on non-target insects. This is especially desirable when beneficial insects are being conserved to aid in an overall integrated pest management, that is IPM, integrated pest management program, or when an ecologically sensitive area is being treated. Let's move on to the next question. Organic farming does not include green manures, chemical fertilizers, farmyard manures, or compost. And the answer for this is B, which is chemical fertilizers. Let's know how. Organic farming includes several methods to enhance soil fertility, matlab to increase the fertility of soil. Fertility of what? Soil. In such farming methods of biological origin are used. For example, biopesticides, biofertilizers, IPM, which was integrated pest management program, green manure, bioherbicides, etc. Chemical fertilizers are not used in organic farming. Chemical fertilizers are not used in organic farming. And let's move on to the next question. Cyanobacteria are heterotrophs and chemotrophs or autotrophs or cyanotrophs. And the answer for this is autotrophs. Let's know how. Let's know first what are autotrophs. Cyanobacteria are photosynthetic gram-negative prokaryotes. They have chlorophyll A and are probably manipulative major primary producers in the world's oceans. Hence, they are autotrophs. Symbiotic nitrogen-fixing bacteria form a mutually beneficial association with the plants. So the bacteria obtains food and shelter from plants. So then in return they give a part of their fixed nitrogen to the plants. The most important of the symbiotic nitrogen fixing is rhizobium. It is rhizobium. So it forms nodule of the roots of legume plants. They develop the ability to fix nitrogen only when they are present inside the root nodules. In the nodule cells, bacteroids lie in a group surrounded by the membrane of the host, which is lined by pink and red pigment, which is called as leg hemoglobin. Now, what is surrounded by membrane of the host, which is lined by pink and red pigment, which is called as leg hemoglobin? The nodule cells, the bacteria which lies in the group. Let's move on to the next question. A nitrogen fixing microbe associated with the fern azolla and rice fields is Frankia, Rhizobium, Spirulina or Annabina? And the answer for this is Annabina. Let's know how. Okay, let's come back to the answer. Annabina azola. Annabina azola, a cyanobacterium that lives in symbiotic association with the free floating water fern, which is azola. So, Annabella, not Annabella, Annabella azola can grow photoautotrophically and fixes the atmospheric nitrogen. The inoculation of cyanobacteria in rice crop significantly influences the growth of rice crop by the secretion of ammonia in food water. Nitrogen fixing cyanobacteria, which is also called as blue-green algae, forms symbiotic association with several plants. For example, Annabina, nostoc, association is of great importance to agriculture. Azolla pinnata is a free floating fresh water form which multiplies rapidly, doubling every 5 to 7 days. The fern can coexist symbiotically with Annabina cyanobacteria and fixes the atmospheric nitrogen in paddy field for rice plant, and it does not interfere with their growth. But in some Southeast Asian countries, especially China, the rice fields are regularly provided by Azolla bacterium. Okay, so let's get the pure concept of this. Nitrogen fixing microbe, which is associated with the fern azolla in rice fields. In rice fields is Frankia, Rhizobium, Spirulina or Annabina. Now, Annabina azolla is association with of great importance to agriculture. Azolla pinnata is a free floating rice field structure and fixes nitrogen in paddy field for rice plant, and it does not interfere with their growth. In some Southeast Asian countries, especially China, the rice fields are regularly provided with azolla. Now let's get the concept of IPM. Now what is IPM? Integrated Pest Management. What is IPM? IPM involves use of different pest control methods which are ecologically several, that is not cause hazard to environment. Example, biological control methods, better agricultural practices like crops rotation, sanitation, etc. And the most important example is the soil bacterium, that is BT, which is Bacillus thuringiensis. Spores of this bacterium possesses the insecticidal cry protein. So therefore spores of this bacterium kill larvae of certain insects. The commercial preparation of the B. thuringiensis contains a mixture of spores, cry protein, which is also called as B. thuringiensis, cry protein and B. thuringiensis. Now let's know what is alcohol fermentation and SCP. Single cell protein is a production of microbial biomass, spirulina, yeast, Fusarium graminearum. SCP is useful reducing environment pollution by managing industrial agricultural wastes. Now, as we know that there are many soybean and wheat products. Let's know which are they in different countries and named as what. So other foods like tempeh. This sounds a bit funny. Tempeh, tofu, which we all know. It's like paneer, soyabean paneer, which is also called as, which is very proteinous and mineralized. Tofu की बात sufu. Sufu is Chinese. This is from Japan. This is from Indonesia. So antibiotics are organic compounds produced by a microorganism in low concentration, which inhibits the growth or kills the pathogenic organism without harming the host. Antibiotics are obtained from bacteria and fungi. So we got this answer. The nitrogen fixing microbe associated with the fern azolla in rice fields is Anabaena azolla. Anabaena azolla, nostoc, oxyclitaria. Oxyclitaria looks like this way. Kind of earthworm and snake. Yeah. I think nostoc, oxyclitaria, Anabaena, and Azulobacterium is what? Azulobacterium is used especially in China. For what reason? The rice fields is provided by azolla in China. And the last question is, single cell protein can be obtained from bacteria, fungi, algae, or all of these? And the answer for this is D, which is all of these, that is bacteria, algae, and fungi. Let's know how. Now, the cell from microorganisms such as bacteria, fungi, filamentous algae, treated in various ways and used as food, are called single cell protein SCP. The biomass of not only obtained from unicellular organisms but also from multicellular microorganisms.